right, hello everybody. Today we are going to move beyond Mendelian genetics, but beyond simple dominant and recessive traits and mono and dihybrid traits to things that maybe were a little bit more complex that Darwin never even knew about. Let's go ahead and get started. First thing we're going to talk about, I always teach these simultaneously, is codominance and incomplete dominance. In both circumstances, we have two genes that are equally dominant. In the case of codominance, if somebody inherits one dominant trait from one parent and a different dominant trait from the other parent, both genes show up in the phenotype, what they look like. So heterozygotes experience both of the traits at the same time. The classic example is blood types. Type A blood is dominant and produces an A protein on the surface of blood cells. The type B allele is also dominant and it produces a B protein on the outside of red blood cells. So if somebody gets an A protein, an A allele from one parent and a B allele from the other parent, they have type AB blood, and you can see from the diagram, it means they get both the A and the B proteins on the surface of their red blood cells. Another example are cattle. There's red cattle, which are kind of a ruddy brown color, and white cattle. Both of those are codominant. So you show them with a C, it's a C for color, and then a superscript capital W or capital R. And so if it's a white, it's CW, CW. And if it's a red, it's CR, CR, it's capital R, not lowercase r. But then if they have offspring, the offspring will be CW, CR, and they are called roan because they have both red and white fur. So that's an example of codominance. Incomplete dominance is kind of what people in Mendel's era believed usually happened, and that's the blending idea. So that's where they're both equally dominant. You can see it in this diagram. Let me see if I can, in the Punnett square here, I'm going to get my picture out of the way. So it says in heterozygotes, again, a third blended phenotype is expressed. So in this case, if an individual, a flower, a snapdragon, is CRCR, then it's red. If it's CWCW, then it's white. But if it's heterozygous, it blends the two colors and we get pink. Instead of, if this had been codominant, we would see both colors and the CRCW would be red and white. In this case, it's blended to be pink. All right, the next one that I always teach together, the next two that I always teach together are multiple alleles and polygenic inheritance, because both multiple means more than one, and poly means more than one. They're concepts that get kind of turned around in students' heads. So let's start with multiple allele. Blood types are, again, a classic example of multiple alleles. Multiple alleles mean that they have more than two choices for a single gene. So in the case of blood types, there's type A, a codominant trait or codominant allele, type B, also the codominant allele, or type O, which is a recessive trait. So that's three different allele choices for just one single gene that determines blood types. So anytime that we have more than one allele, that's called multiple allele. And then, of course, we said blood types are examples of both multiple alleles and codominance at the same time. Would you take a moment to just notice how blood types are written? Because sometimes that can be a little bit confusing. I would like to just write it capital A, capital B, and lowercase o, but that's not how they do that. So they do a capital A with a capital I with a superscript capital A or capital B. Those are for the codominant alleles. And then for type O, it's a lowercase I, no superscript attached to it. So just something to be aware of. So if you see that, which you will, you're not confused by it. Now, polygenic, the prefix poly means many, but this time it's many genes. Multiple alleles was about many alleles, many versions of a single gene. Polygenic is about many genes working together to produce a whole continuum of phenotypes. So it says when there is more than one gene that contributes to the phenotype. Examples: skin color, eye color, height. There's many, many, many examples. Hair color. Instead of just brown skin or not brown skin, we have all these shades of skin color. Instead of tall or short, we have all these different variations in height. Instead of blue or brown brown eyes, we have a myriad of different eye colors. And that's because there's a whole bunch of genes that are working together. And this Punnett squared begins to give you a tiny sense. I read in the article that's linked here where it says many genes, I read in the article that for a while we were thinking that skin color was controlled by something like 100 different genes. We've changed that a little bit as we've learned more about skin color, but we know that it's being controlled by more than just one gene, which is what Mendel, when we're working with Mendel, it seems like everything's one gene and it's dominant or recessive, and it's way more complicated than that. All right, so now let's take a look at analyzing pedigrees. We're going to do a whole section later on in the course where we actually look at pedigrees and not only determine the genotype of each person that's in the pedigree, but also determine, is this a sex-linked recessive trait? Is this an autosomal dominant trait? And we'll get to that, but not quite yet. So what is a pedigree? A pedigree is a family tree. You may have heard of the word pedigree in reference to dogs. And when we talk about a dog's pedigree, we're talking about their ancestors. So if you have a purebred dog, we might want to prove that they're a purebred dog, and you do that with a pedigree. So on these family trees, the males are shown with boxes and the females are shown with circles, biological males and females. They're colored in, and that indicates when they're colored in that they have the trait. So in this pedigree, red is colored in and the blues are not colored in. So the red individuals have the trait. So then if we take some time and look at the pedigree, it looks like we have 4 generations of families here. So we start out with the great-grandparents up at the top. We've got a male and a female. The female has the trait. Then they have, now be careful, look at it carefully, how many children they have. They have a red female and then a red male. So they both have this trait, whatever the trait is. We don't know if it's dominant recessive yet because we haven't analyzed it. But then their red male apparently had children with a female, and so they had some kids also. But notice that blue female, that she doesn't have a line going up attaching her to the other siblings. So she's married into the family, or she has mated into the family, but she's not part of the original grandparent family or great-grandparent family. Okay, and then moving one over, we have a male that is unaffected. One more, we have a female that's unaffected that had children with a male. And then finally, at the very end of that second row, we have a female that is affected. So you can start looking, we've got 4 generations and who's been impacted and who has not been impacted. So something kind of interesting or fun to do with the pedigrees is to determine, is this trait due to a dominant or a recessive allele, or could it be either one? Sometimes you can't tell just by looking at the traits. So let's figure it out. So let's do some examples. So if this is a pedigree for a recessive trait, how would that look? So everybody who is colored in, the only way that they could be showing the trait is if they have 2 copies of the recessive trait. So they would be little r, oh, I'm sorry, I don't know where I got the letter R. Let's use the letter N. Actually, I like to use the letter N. N means no trait or normal. And so that would be capital N. And then lowercase n means they have the recessive trait. So let's use the letter N. So everybody who is red here would be little n, because they don't have the dominant trait, and we're saying that it's a recessive. We're experimenting. We're thinking, could this be a recessive trait? If it is, everybody who's red is little n, I hope I explained that well. Okay, so is it possible that a couple where the dad does not have it, I'm looking at this generation right here, point to it right there, the dad does not have it, but the mom does, could they have a combination of kids who have the trait and kids who do not? And the answer is yes, if dad is a carrier or a heterozygote. So we know that dad is capital N if this is a recessive trait because he's normal, he's not colored in. His second letter could be another capital N. He could be homozygous dominant. If he were homozygous dominant, then they could not have any kids that have this trait because he would always pass on one of his dominant traits. And so all of the kids in the second line would be blue. So could this still be a recessive trait though? It could be, as long as he is heterozygous. If he is capital N, lowercase n, then he can pass on a recessive trait to his children, and the female can pass on the recessive trait to her children, and then we would have some that are dominant and some that are recessive. So it does work, and if we follow the whole line of thinking through, it is possible that this is a recessive trait that we're looking at. So now, one thing I will mention, this is a rule of thumb and it's not a guarantee, just helping you in analyzing. Recessive traits often, not always, recessive traits often skip generations. And that's because, especially when we're talking about genetic disorders, they're fairly rare. So if everybody had the recessive trait, then okay, you're going to see lots and lots and lots of people with the recessive trait. But typically, when we're talking about genetic disorders, it's fairly rare. So it's going to vanish in the second generation. And it requires two people who are heterozygous to come together, and then they have a baby that has it. So that's not a for sure, but just an idea. So this one didn't skip any generations, and that's more indicative of a dominant trait. So let's see if it could be a dominant trait. The other thing that I'll just mention I notice is that it's affecting males and females pretty equally. So I would say that this is not a sex-linked trait. Remember that sex-linked traits, actually we'll come up to that later, sex-linked traits are more likely to affect boys than they are girls. So we'll come into that. Okay, so could this be a dominant trait? Well, let's use capital N this time. Let's use capital R. I'm going to change the letter. Let's use capital R for red. And we're going to say that red is dominant. So the female has at least one capital R. She could be big R, or she could be big R, little r. I will tell you as an aside that in genetic disorders, usually having two copies of a dominant disorder is not functional. So that means that those people either don't survive, or they're less likely to have offspring, or their disease is more severe. So for the most part, we're not going to see very many dominant genetic disorders where somebody is homozygous. Occasionally, but not very often. So she could be, though. So she could be big R, or she could be big R, little r. The husband or the male has to be little r, because he's not showing this dominant trait. So could they have some kids that don't have it and some kids that do have it? Well, yes, they could, because if he's little r, and if she happens to be big R, little r, which is more likely, then they're going to have a mix of kids who have the trait and kids that don't have the trait. So as I look through, it still looks like this is a possible dominant trait also. So in this case, it could be a dominant or recessive. There are things that give us a hint that it's probably dominant because it's showing up in every single generation. That makes it more likely, but it could be recessive. All right, here's another example of a pedigree I just wanted to show you. Sometimes in pedigrees, like with codominant or incompletely dominant pedigrees, or just pedigrees where they happen to know for sure that somebody is heterozygous. Remember the other one, we weren't sure necessarily if they were heterozygous or not, like we could make some guesses. Sometimes they show it as a box that's partially filled in. So it'll be a little bit of red or a little bit of black and white or something like that. So here's an example. Sickle cell anemia is incompletely dominant at the cellular level. Now, this might be confusing, but just listen, because heterozygotes are producing some sickle-shaped cells in low oxygen environments. So they are incompletely dominant. That means that they have they have two of the traits. They have the sickle trait and the not sickle trait, and they're blending together so that that person produces some, but not all of the sickle cell shapes. Normally, we think of sickle cell anemia across the board. We normally think of it as a recessive trait. So it says, which of the individuals on this pedigree are producing limited numbers of sickle-shaped red blood cells? And it would be any of the ones that are red and blue. Those would be the heterozygotes. We'll talk more about pedigrees as we go. We got to get a little bit more information. All right, non-nuclear inheritance. For the most part, in a genetics class, we talk about the nucleus and we talk about our DNA. But we learned a while back that mitochondria and chloroplasts have their own DNA and that they replicate on their own. So there are traits that are carried in the mitochondria that get passed on. And interestingly, they only get passed on through the mother. Sperm is incredibly tiny, and it's basically a packet of DNA with a tail on it. Like, that's about all sperm is. The egg is enormous in comparison to a sperm that would be about that big. And the egg is the thing that carries all the organelles. So the egg is the one that carries the mitochondria. So because the egg carries the mitochondria, all mitochondrial DNA comes from the mom. So we can follow mom's line of the family by taking a look at the mitochondria. What might be surprising to many of you is that there are hundreds hundreds of mitochondrial diseases that affect human beings because something's wrong with the DNA, not of their nucleus, but of the DNA of their mitochondria. All right, environmental influence. The interaction of an organism's genome, that's the word, the genome means all of their DNA, and its environment leads to complex patterns of inheritance. These are just some classic examples, and there's some super simple examples. Hydrangea flower color, maybe some of you have seen hydrangeas. Depending on the acidity of the soil, that for some of them will determine whether they have blue or pink flowers. You're all familiar with Arctic foxes and Arctic hares, and there's many different animals that change color. So their hair coat color depends on photoperiod, not temperature. A lot of people think it's determined by temperature. Photoperiod means it's determined by how much light they get. So that works well because when there's less light, there's more snow, and when there's more light, there's less snow. So their camouflage works out perfectly that way. Global warming could screw that up. Take a moment to think about why that would be. All right, other examples. Plants. Who knew plants can respond to herbivores by changing their, so they have different defenses against herbivores like thorns and hairs and chemicals that taste bad and chemicals that make them itch like poison ivy. So the more that they experience herbivory, which means predators that are eating them, they actually are able to respond genetically and produce more thorns or longer thorns, distasteful chemicals, more trichomes, whatever. And so that's what these two graphs are taking a look at. Fascinatingly, the picture down below, and some of you may have heard of this, the temperature at which turtles, crocodiles, and some and American alligators, the temperature at which their eggs are incubated determines the sex of the babies. So what do you suppose global warming does to that? We're going to learn a little bit more about that later on, but super interesting business. Okay, and then epigenetics. Let me get my picture away from the mammogram here. So epigenetics is something that we don't talk a ton about in this class, but you will have, if you decide to go into biology, you'll probably have an entire class on epigenetics. Epigenetics is the study of the gene and environment interactions in gene expression. So epigenetics alters the way that genes are read and expressed, but not the genes themselves. So when we talk about epigenetics, we're not talking about mutations. That's regular genetics. When we're talking about epigenetics, we're talking about things in the environment that are affecting whether our genes turn on or off. And those are things that carcinogens that we could come in contact with, all kinds of things that can either turn our genes on or off when they're not supposed to. So here's an important example. The BRCA1, that's a 1, not an I. The BRCA1 gene is a tumor suppressor. So we know what tumor suppressors do. BRCA1 is regulated by something that's called microRNA. And we're going to learn about microRNAs in our next unit, but just know that it regulates it. It turns out an epigenetic change to the microRNA that's in charge of the BRCA gene, if there's changes to the microRNA, it decreases the BRCA1 activity, so it's not tumor suppressing the way it's supposed to, and that can lead to breast cancer. So that's super important, and we'll learn a little bit more about that as we go, but that's what epigenetics is. And then phenotypic plasticity. I got this diagram straight from the Biozone ebook. I really like it. I've got a quote here. For many phenotypic traits, so what they look like, that kind of thing, especially those controlled by multiple genes, I'll come back to that, organisms may show varying degrees of flexibility in their observable phenotype. So all kinds of things, and this diagram is just showing some of the things that affect what the organism is going to look like. Anything from their genotype to their epigenetics to their environment. Those all are things that have an impact on their phenotype, what they look like. Okay, so let me get out of here for just a second. So I said I was going to mention the significance of multiple genes. So variation, you can have blue or red flowers, and there's just two choices and there's not a lot of variation. We call that discontinuous. Or you could have an example like height, where you have somebody who's 4'6", 4'7", 4'8", 4'9", all the way across. More and more people in the middle, less and less people. That's continuous. Where you have a large number of phenotypic variants, that's usually determined by multiple genes, as height is. And it's usually heavily influenced by the environment. So an example of how height is influenced by the environment is a lot to do with nutrition. So maybe many of you have heard that way back in the day, I don't know, I don't have a date, that humans were shorter generally. And that was because we weren't getting as good nutrition. Now, of course, we have obesity and we have diabetes now, but we're generally not vitamin deficient. We're eating too much, but we're generally not vitamin deficient in the way that we were in the past. So we're achieving more and more of our maximum height instead of in the past where people were shorter. So it says if there are multiple genes, it's heavily influenced by the environment, so it's more likely to show phenotypic plasticity, lots of flexibility in the phenotype. It's not just one or the other. Discontinuous, that's what I was talking about with like a red flower or a blue flower. Limited number of phenotypic variants. It's usually determined by just one or two genes, and that's less influenced by the environment. And then if you just take a look, this is the very last thing, and then we're done. Take a look at these butterflies. They're called buckeyes, and take a look at the graph because it's kind of interesting. The buckeyes, their color changes depending on the temperature of the environment. And so it turns out that low temperatures cause the buckeye butterfly to become darker, because when they're darker, maybe you've experienced this in the winter, that absorbs more heat from the sun and it warms them up. So if you wear, for example, a dark jacket on a cold day and you're out in the sun, you'll feel warmer than if you wear, say, a white jacket on a cold day and the sun hits it. The sun reflects off a white jacket. All right, so those are all examples of phenotypic plasticity. The last 2 pages are just things that you really need to know, and I'm not going to go over those. You can read them on your own. Have a great rest of your day.